Welcome to The Mind, the Cure Protocol for July 30th, 2026. We begin with what changed, not merely what was announced. One movement in this path has already reached care. The rest are research signals that could open the next path, but each still needs proof. We will start close to real people, then trace the evidence back to the targets and questions that matter next. This protocol is a living loop. People bring the questions that matter. Direct evidence changes the map. The source ledger remembers what we have already seen and checks the original date before calling it new. Every signal ends with one practical next action, the experiment, validation, collaboration, or care decision that could move it closer to real benefit. AbbVie announced European Commission approval of RINVOQ, upadacitinib, for severe alopecia areata in adults and adolescents, based on Phase 3 UPAA data showing statistically significant scalp regrowth and eyelash improvement, with first-in-class JAK inhibitor achievement of complete scalp hair regrowth versus placebo at week 24. For people within the stated indication, this is now a regulated care option. Treatment decisions still belong with patients and clinicians. Approval establishes regulatory status for the stated indication, not proof of superiority or the right choice for every person. Next, what real-world uptake, safety, and outcomes within the approved indication? Researchers in the MAIA-FLAME 19 multi-center, randomized, double-blind trial reported in Nature Communications that Losartan plus prednisolone did not improve the primary MRI measure of myocardial inflammation in 279 patients with post-COVID cardiac symptoms, despite targeting suspected immune and endothelial dysregulation. The result matters because it shows how AI-driven and data-rich disease research is increasingly treating syndromes like post-COVID as an engineering problem, identifying a measurable failure mode, testing a causal intervention, and using negative results to refine the path toward a true phase transition from symptom management to mechanism-level cure. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. Researchers report Raygun, a generative AI framework that uses protein language model embeddings to miniaturize, modify, and augment natural proteins while preserving function, addressing a long-standing gap between sequence design and nature's own evolutionary editing. The result is significant because it shifts protein engineering toward an intelligence inversion model. Instead of discovering disease interventions by slow experimental iteration, AI can now rewrite biological molecules as if they were engineered systems, pushing medicine toward a phase transition in how cures are designed. If confirmed, this could help drug teams test better molecular ideas sooner instead of searching blindly. The useful part is a smarter starting point, not a finished medicine. Biology still gets the final vote. Next, show with independent benchmarks and laboratory work that the targets produce better drug candidates. Researchers publishing in Nature showed that senescent cells execute the SASP through a two-layer control system. Mitochondrial nucleic acid signaling initiates inflammatory transcription, while an upregulated mitochondrial pyruvate citrate acetyl-CoA axis supplies acetyl-CoA to drive histone acetylation and robust SASP gene expression. This matters because it turns senescence from a vaguely SAGE-related state into a tractable engineering problem with a defined metabolic epigenetic control point, illustrating the intelligence inversion at the heart of AI-enabled biology. Once the causal circuitry is mapped, disease becomes an optimization and control problem rather than a descriptive one. If this holds up, it could give researchers a clearer target for the next experiment, validation step, or collaboration. The useful part is a clearer target, not a finished solution. Independent evidence still has to do the hard work. The next question is whether independent evidence confirms the finding and shows a responsible path toward real-world use. The practical read is simple. These are signals, not promises. But each one is a concrete target that researchers, clinicians, engineers, funders, and the people most affected can connect to a next step. Keeping the evidence visible is how the cure protocol stays current without pretending the work is finished.